showed you this slide last class, and this is showing an exergonic reaction. Specifically, it's showing us cellular respiration. And aerobic cellular respiration, you can do the glycolysis part of cellular respiration, and I'll show you that reaction today, without oxygen. But if you want to do the full cellular respiration, all the different processes of it, you have to have oxygen. So that's why it says aerobic. And what we're doing in cellular respiration is we're doing an oxidation reaction. So glucose gets oxidized. Now, what it means to be oxidized is you're losing electrons. Basically, there are all these electrons that are in this covalent compound here, and we're going to strip away those electrons from glucose. And when we do that, we will then release a bunch of energy. How much energy is your delta G, which is a negative number, and then that energy release, we can fuel that to make ATP. Okay, that's kind of like the big picture view of what we're trying to do in cellular respiration. What this slide specifically is also trying to show is this activation energy. So even though like this reaction is exergonic, if you had a fuel like a carbohydrate-based fuel, or basically any fuel that has a lot of carbon to hydrogen bonds, those bonds, because this is more of a non-polar bond, the electrons are not like pulled. The electrons are more like equally shared in this bond. And that is what allows us to be this is what makes this a good molecule to oxidize, to remove those electrons. Anyways, you might be asking, well, when we do this, how come this reaction doesn't just happen why doesn't like the gas in your car just like explode all at once? And the reason is activation energy. So the idea of cellular respiration is, and I'm going to show you all these enzymes that we need to lower this activation energy so that this reaction can actually happen in an efficient manner. Okay. I got some other summary slides from last class, so I just wanted to kind of go over pretty quickly. As I said, the idea of what we're doing in cellular respiration is we're stripping away, we're oxidizing our glucose. And the purpose of that is to remove electrons, and then we want to shuttle those electrons to the electron transport chain. That's what we're going to talk about on Thursday. Electron transport chain. And so the question is, well, okay, maybe you understand how you could remove the electrons from glucose, but how do you get the electrons from glucose to then make ATP, or get them to the electron transport chain? That's where nicotinamide adenine dinucleotide comes in. NAD comes in here. So breaking down like what this molecule is, nicotinamide, that is referring to this part specifically. Adenine, that's referring to the adenine nitrogenous base. Nicotinamide is also a nitrogenous base. These are both, I can label that for you. These are both nitrogenous bases. You saw nitrogenous bases when we were going over nucleic acids. They were made of nitrogenous bases, so your DNA and your RNA. And then dinucleotide, di because there's a nucleotide. Remember the Tide Pods, there's 3 colors. A nucleotide is when you have the nitrogenous base. Your sugar, in this case this is a ribose sugar. Here's another ribose sugar. And then you have 2 phosphates. So together, this plus this plus this, that's a nucleotide. That plus that plus that, that's another nucleotide. There's 2, so di. So there you go. It's not just, I know it sounds like super intimidating when you see the name kind of spelled out like that, but it's named in a way that actually does make sense if you can understand like some of the basic chemistry of this. Anyway, so we take those electrons from glucose, and what we're doing, we are going to attach those electrons to this NAD+. So as it's shown right here, this is NAD+. That's what they're trying to show by putting that nitrogen in blue there. So what we do is we attach those electrons to this to the NAD+, forming NADH. So, and whenever you're attaching those electrons, what that practically looks like is the addition of a hydrogen atom. So basically, we're gonna attach another hydrogen bond to this N plus. And by doing that, that would be the same thing as reducing this molecule. Reduction is gain. So this is the molecule gaining electrons, so it's going to lose that positive charge. And in the process, we're going to release some H pluses. Those H pluses will then feed into, they'll be a part of the electron transport chain in this process called oxidative phosphorylation, where we're going to use those H pluses to then make the ATP. Okay? So NAD plus, it's an electron shuttle. Another kind of electron shuttle is also FAD. I'll show you FAD when we go over 7.3. It's another electron shuttle. All right? And then I showed you this slide here where, this would be like the fuel in your car, where like if you just like strip away all of those electrons, if you oxidize glucose all at once, you can release all that energy, that delta G of that exergonic reaction, but then it's like all the energy is released at once, and it's hard to really like utilize it, so it's just going to be released as like heat and a bunch of light. The idea of that electron transport chain is we're going to take those electrons from glucose. We take those electrons from glucose, we transport them via NADH and the FADH2 to the electron transport chain, and then we do like a stepwise controlled release of that energy to make ATP. Okay? So that's what I want you to get out of that for now. All right, and this was your kind of overall big picture of cellular respiration. So you have glycolysis. Glycolysis, I'll show you the details of it. Basically, you're going to take glucose, and you begin the oxidation process where you're going to break it down into pyruvate. Glucose is a 6-carbon sugar. You basically take glucose, a 6-carbon sugar, and make 2 3-carbon sugars. These 3-carbon sugars are called pyruvate. And then you're going to take, and this happens in the cytosol. So you need to know, if you're like, all right, what test question-wise, you need to know inputs, outputs, and where it occurs. That's really most of the questions you're going to see. So the quizzes is going to be pretty helpful for that. So it's kind of a lot of, it's pretty fact kind of based. Meaning when I show you a lot of the reactions in detail later on, if you take biochemistry in college, you need to know those, like the actual specific reactions. Here it's more of understanding this overall inputs, outputs, and location. Okay. Anyways, we make a little bit of ATP. I'll explain substrate level phosphorylation, I think on the next slide, what that is. It's a pretty simple idea. Then you take pyruvate, you put it inside the mitochondria, and you do pyruvate oxidation. So you're doing another oxidation reaction where you're going to move even more electrons, and you're going to form something called acetyl-CoA. This is going to be a 2-carbon sugar. And you take that 2-carbon sugar, you put it into the citric acid cycle, and you oxidize it even further. That's why you're having even more electrons coming out of, pyruvate oxidation and the citric acid cycle. All of those electrons then get fed into the electron transport chain, where we make the most amount of ATP. That's why they have like the thingy around the ATP bigger. You make the most amount of ATP here. You only make 2 ATP, but here you make 32 ATP. 2, 2, 32. It's like a rap song. It's like a rap beat. 2, 2, 32. 2, 2. Or not. Okay. All right. Anyways, all that's going on in the mitochondria. All right, I'll keep it moving. As I said, oxidative phosphorylation, 90%, so 32 ATP is the number most commonly stated in that oxidative phosphorylation. And then, in oxidative phosphorylation, what you're actually doing is you're taking inorganic phosphates and you're reattaching them to ADP to regenerate that ATP. I'll show you that reaction in more detail when we actually do that on Thursday. All right, substrate level phosphorylation. So substrate phosphorylation is how we make ATP and the glycolysis and the citric acid cycle. So I want to go over just really briefly what that process is. Basically, you have a substrate that has a phosphate group. You have an enzyme that will catalyze this reaction. It'll catalyze the transferring of the phosphate to your ADP to make ATP. And then the ATP in your product, which is the substrate that lost the phosphate, will leave the enzyme, and then that enzyme can then catalyze that reaction again. So that's it. It sounds a lot scarier than it actually is. The substrate is doing the phosphorylation. So the name kind of gives away what it's doing. Okay. Yeah, for each molecule, we make up to 32 molecules of ATP. You'll see that number change. Sometimes I've seen the range like 32 to 36. The 36 is what I was assuming when I showed you 32. I'm not like nor would I can imagine the AP exam like really like hammering you on that number either way. It's more of understanding that the oxidative phosphorylation, what we do here, that's what makes the most amount of ATP, and you only make 2 in the substrate level phosphorylation. Okay. In other words, sometimes you make only 28 ATP here, but you always will make 2 ATP in the substrate level phosphorylation. Okay. All right. I think now that leads us to 7.2. So getting into glycolysis. So in glycolysis, we're going to oxidize glucose to make pyruvate. Glycolysis, what it literally means is sugar splitting. So what you're doing in glycolysis, you're taking glucose, which is a 6-carbon sugar, and you're going to split it into 2 3-carbon sugars. Those 3-carbon, what? Those 3-carbon sugars we call pyruvate. And then in the process, you'll also release 2 water molecules. Sometimes when you see like, I say you know like the inputs and the outputs. Sometimes you won't see water as an output if you look at like the charts. No 2 water molecules is a product. Especially for like the AP exam, like some people get more particular about like technically water's produced here. So just know that. Know where it occurs. It's occurring in the cytoplasm, and this is for all cells. Whether this is a bacteria cell, a eukaryotic cell, it's always occurring in the cytoplasm. And then understanding, you don't need to know like I'm going to show you the actual reactions here. You don't need to know the details of these reactions, but you have to understand there's an energy investment and an energy payoff phase. So the first 5 steps are the energy investment phase. And then the last 5 steps are the energy payoff phase. Really just understand first half you invest energy, second half you get the energy back. Okay? So what's going to happen here when we invest energy, we're going to invest 2 ATP. You put 2 ATP in, you get out 4 ATP. You spend $2 and you make $4. So you only net $2. You only net 2 ATP. Okay? But you will also get out 2 NADH each time you put a glucose molecule in there. So in other words, another product of glycolysis, that the products of glycolysis, 2 water molecules, 2 ATP, and 2 NADH. Think 2s. 2, 2, 2. Right? What's what? Is it really? 2, 2, 2? Plus 4, 4, 4. Drake guy, he gets it. I don't know about the new Drake album though.
You don't like it?
Not really. Hot take.
That's a super hot take.
It's super hot take? I don't know. 21. Okay. Glycolysis occurs whether or not oxygen is present or not present. Meaning, whenever we go over like fermentation, you only do glycolysis when we're doing fermentation. There's some other things that will happen after that to like process the waste. But if oxygen is present, then we can do the oxidative phosphorylation. When they say oxidative phosphorylation, that's where we get into like the electron transport chain portion of things. Okay, this is where you do the controlled release of all those electrons that you get from the NADH. Okay. All right. So let's go over. Or here's another, just kind of overview slide. So we're here. We're in the cytoplasm. It's okay. Now, here's the energy investment phase. You can sharpen, it's fine. I already showed it to you. You take glucose, you invest 2 ATP. Remember, when I say invest 2 ATP, you also then create 2 ADP. So I think maybe in Mastering Bio, they had for outputs like 2 ADP plus maybe 2 inorganic phosphates. That's pretty in the weeds, but it's like, yeah, technically you could consider that a product, I guess, is that you're making 2 ADPs. Okay. Then the payoff phase, you get the 4 ATP, the 2 NADH, and the 2 pyruvate. It's actually 2, 2, 2, 2, 2. Right? 2 pyruvate, 2 waters, 2 ATP, 2 NADHs. And you get really crazy, there's 5 2s. There's 2 hydrogen ions that are also produced. Because water is H2O. 2 hydrogens, 1 hydrogen goes to the NADH, gets attached to the NAD plus to make NADH. The other hydrogen gets released. Those hydrogen ions will then be used in oxidative phosphorylation. So those hydrogen ions then get fed here and the oxidative phosphorylation. Okay. Okay. Oh, I posted in Schoology the notes I used for like freshman bio for like basically this whole unit. I don't know. It just might be kind of useful to kind of see like, well, what did you learn at the freshman level, and then maybe if you're having a hard time like kind of processing all that's happening, maybe it might be helpful to read through those first, and then you go into this level of detail. So, anyways, if it's helpful. All right. So now we're getting into I'm going to show you the actual 10-step reaction process that is glycolysis. When I first met my wife, I was taking biochemistry. In biochemistry, you quite literally have to memorize all these reactions. We would have we had a test, and it was just a blank piece of paper, and it was draw glycolysis. And you'd have to draw the glucose molecule. You would have to know, okay, that oxygen goes down, this one goes up. You have to know the enzymes, know this becomes this, write the names. And so it was just a lot of memorization and just practicing writing it out. And so my phone background was all of glycolysis. And I was on a date with my wife, and I got notification. She was like, "What is your phone background?" I was like, "That's glycolysis." And she was an accounting major, so she didn't really. She was like, "Oh, okay." I got to memorize it for a class. And I don't know if that helped me come off any better, but anyways. All right. So I guess my point is just kind of don't get lost in the details here. I'll kind of explain to you what's going on. In glucose, this is where we're going to invest that first ATP. We're investing ATP, and the purpose of that is to attach this phosphate group there. Okay? The enzyme that helps us do that is called hexokinase. Kinase enzymes, they're kind of a class of enzymes that their main job is to attach phosphate groups. So that's kind of a common theme you'll see. And you then make glucose 6-phosphate. So track with me here. The phosphate, glucose 6-phosphate, there's the phosphate. What do you think the 6 is referring to? What is it? Not the shape. But it kind of does refer to the shape. 6. The 6 is referring to, this is the number 6 carbon. You number the carbons 1, 2, 3, 4, 5, 6. So that phosphate is attached to the sixth carbon. Again, don't have to know that necessarily, but I think it's just kind of interesting to sort of start building that knowledge of what's going on here with the reactions. Okay. All right. Then moving on to the next slide. We have our glucose 6-phosphate. We're then going to take this glucose 6-phosphate and we're going to convert it. We're going to convert this glucose to a fructose. So glucose is a 6-carbon ring. Fructose is a 5-carbon ring. Okay? And that's it. So glucose 6-phosphate to fructose 6-phosphate. The enzyme that does that is phosphoglucoisomerase. An isomerase enzyme is an enzyme that does stuff like this, that kind of does this rearranging process here. Okay? Cool. Now, here is our second ATP. So this is why we're still in the energy investment phase. We're going to invest our second ATP. And where you're going to invest that second ATP, bless you, is to the 1 carbon. So that's why it's fructose 1,6. The bisphosphate, I don't really know what the bis means, but the bi I think is referring to 2 because there's 1, 2 phosphates. Or at least that's what I tell myself. Fructose 1,6 phosphate. So there you go. Fructose sugar, fructose on the 6 carbon and the 1 carbon. Cool. Oh, and then kinase. As I said, kinase, that's an enzyme that specializes in attaching phosphates. So that's why that's the name of that enzyme. And phosphofructo, because it's referring to the phosphate and the fructose sugar. So this seems very intimidating to I have to memorize this one day if I keep studying biology. But it's not as bad as it seems. It comes along a little bit quicker once you understand some of the basic themes here. All right, now we're going to take that fructose 1,6 bisphosphate, so we're right here. And now we're going to do something called aldolase. Aldolase, what it's going to do is it's going to chop this. I told you how we took in glycolysis, take a 6 carbon sugar, and we make 2 3 carbon sugars. This is where, why do I keep writing 36? This is where that process starts to happen. The aldolase is going to chop this 6 carbon sugar into 2 3 carbon sugars. One of them is called glyceraldehyde 3-phosphate. Anybody remember glyceraldehyde? What does that remind you of from Unit 1? Glycerol? Where did you hear glycerol from? No. Not carbohydrates. Fats. Fats. So fats are made of what?
Protein.
No, glycerol plus the
Fatty acid tails. Yeah.
Oh, yeah.
So if you look carefully, this is basically a glycerol molecule with a phosphate attached to the 1, 2, 3 carbon. Glyceraldehyde 3-phosphate. Aldehyde. An aldehyde is when you have a carbon. That's what the C double bonded O H means. Anyways, just talking shop, right? And we also then make dihydroxyacetone phosphate, and that's just kind of an awful name, and there isn't really much to connect that to. But what is important is this dihydroxyacetone phosphate. It actually then gets converted to glyceraldehyde 3-phosphate, which they call G3P because that's a lot easier to say. So DHAP turns into G3P, and this happens this is technically a reaction that can go back and forth, but because, think of it like a vacuum. You're going to, as soon as you make G3P, you're going to use it, and that's then going to fuel this DHAP to convert to G3P. So basically DHAP is only there for a short period of time, and it gets immediately converted to G3P to be used to keep being fed into cellular respiration. The rest of glycolysis and all the other parts. Okay, all of that was the energy investment phase. Now we're going to the energy payoff phase. So now this is where you start getting your electron carriers. So here comes NAD+. So now NAD+, it's going to take G3P, and it's going to remove some of those electrons from it. It's going to remove those electrons and attach. How it's going to attach those electrons is in the form of a hydrogen, and then, as I said, one of those H pluses will get removed, will get released, and you do that 2 times. I'm going to say that again because we had a 6 carbon sugar. We made 2 3 carbon sugars. So if you do this reaction once, you would make 1 single NADH. But then remember, this DHAP then becomes a second G3P, so we're doing this process twice. All of these remaining steps, they happen twice. That's why you start seeing all the 2s coming in. You're doing it twice. Okay? Then what that does, you make 1,3-bisphosphoglycerate. Okay, whatever. And then this is where you start getting your payoff here. Now, we're going to use a phosphoglycerokinase. So kinase, something to do with the phosphate groups. Now we're going to take one of these phosphates. We're going to take the number 1 phosphate, and we're going to remove it from the bisphosphoglycerate bless you, and attach it to ADP, making 1 ATP. But there's a 2 there because we do this 2 times, right? Got to do it 2 different times, so you make 2 ATPs. So now you just have 3 phosphoglycerates. So we dropped the biz, right? We got rid of the biz, all right? Got rid of the biz. Now it's just 3 phosphoglycerate. And then you have another, you have phosphoglyceromutase. The mutase takes this phosphate group from the 3 carbon and transfers it to the 2 carbon. So that's why it's 2 phosphoglycerate now. Okay? Almost done. Now we have something called enolase. So enolase, what it's going to do, it is going to basically create a double bond here. It's going to remove a water here. It's going to remove a water from here. That's why you do it 1 time, you're doing it 2 times to make 2 waters. What that does is it creates this double bond right there. Okay. And then the kinase comes in. So kinase has to do with the phosphates. The kinase strips off this final phosphate, creating your second ATP. Cool. And you do that 2 times. And that's the end. So again, don't need to know all the details. I think, excuse me, man, I get choked up when I talk about this. I think it's helpful to kind of know, like, begin to think about, oh, kinase has to do with phosphates, understanding some of like the naming here, just more for your just biological development, you know. But really just understanding inputs and outputs, that's really what you're going to get hammered on the AP exam. Okay. All right, and here's that slide again. So here you invest 2 ATP. These were steps 1 through 5. You then can create 4 ATP, 2 NADHs. So you create 4, but you only net 2. So think of the 5 2s. 2 pyruvate, 2 H2Os, 2 ATP, 2 NADHs, 2 H pluses. All right. Questions here? All right, now that pyruvate, the pyruvate, that's the name of this 3-carbon sugar. This pyruvate is now going to be put into the mitochondria, and we're going to do something called pyruvate oxidation. Pyruvate oxidation is super short. Oh, getting ahead of myself. Let me stop us there.